In this video, we will learn how proton NMR relaxometry can follow the evolution of the different pore spaces during the sealed hydration of white Portland cement. Furthermore, we will look at the density of the CSH formed and its chemical composition. In this video, we will learn how proton NMR relaxometry can follow the evolution of the different pore spaces during the sealed hydration of white Portland cement. Furthermore, we will look at the density of the CSH formed and its chemical composition. We have already seen in other videos that after the inverse Laplace transform of the NMR data, we obtain a dataset looking like this. Each peak represents a distinct population of hydrogens in cement. On the y-axis, the signal intensity is plotted, and on the x-axis, the T2 relaxation time of the hydrogens is plotted. If the hydrogen is bound in water, this axis is an effective pore size. Remember, as you see on the blackboard, the intensity of a hydrogen population is the area under a peak. If the sum of all intensities is normalized to 1, the intensity is a fraction of total hydrogen or water associated with that population of the total sample content. It is important to note that this diagram actually shows the population after 6 days. The first peak can be assigned to the hydrogen bound in the crystalline hydration products shown in orange. In this video, we assume that these are only ettringite and portlandite. The next peak, red, is associated with the hydrogen in water located between sheets of CSH, also referred to as interlayer pore space. Since the water in this space is quite confined, the relaxation time is the shortest of all water populations. The green peak is associated with the water located in gel pores in CSH. In the picture we use here, gel pores are larger cavities with a size of a few nanometers located in defects between extended sheets of CSH. The dotted blue peak is the water located in interhydrate pores. Interhydrate pores are the remnants of the capillary pores which have a constant size. This assignment will be justified during the next minutes. The blue peak describes water in larger pores, such as capillary pores. We have already said that this is the hydrogen population after 6 days of hydration. Cement hydration is a dynamic and complex process, so what happens over time? This is what this video is about. If we go back by 5.5 days, we see that after 12 hours of hydration, the water population looks slightly different. The peak intensities have changed, also the peak positions have shifted. Just after 28 minutes of hydration, the result is drastically different. The spectrum is dominated by the blue capillary water peak and 2 peaks with negligible area in the T2 range, where the interlayer and the gel peaks appear later. The raw data in this graph is difficult to read, therefore, we plot the information in a different way. In the first graph, we plot the intensity of the individual populations against the hydration time, which looks like this. The lines in the different colors represent the solids, orange, the interlayer water, red, the gel water, green, and the water in interhydrate and larger cavities, blue. For the analysis here, this water is always grouped into a single line. After about 2 days, the designation of this population changes to interhydrate pores. We will argue in a minute why. The second graph contains the relaxation times associated to the individual peaks. The x-axis is again the hydration time, whilst on the y-axis the relaxation time of the individual populations is plotted. Please note that this is an effective pore size. Again, the curves are plotted in the same colors as before, but we do not plot the solids since their relaxation time is constant. Again, red is for the interlayer, green for the gel pores, and blue is for the larger cavities and the interhydrate pores. This graph effectively describes how the pore sizes of the different populations change over time. Let's look in detail how these curves develop over time. To make things easier, we hide the curves first. At first, we note that the intensity of the capillary water decreases. We also note that at the same time, its relaxation time decreases. This means that the capillary water is consumed, whilst the pores it is confined and shrink. At this moment, mostly solids are produced and a small amount of gel water. The trends regarding the capillary water and solids continue, but now the interlayer water population increases slightly. The gel pore population increases strongly. Note that the capillary pore size decreases rapidly. Now, after about 2 days, the pores confining all 3 water populations have reached an almost constant size. The gel pore formation continues to be rapid, but now the interlayer formation is almost as fast. Beyond this point, the picture changes again. The first observations we make is that the blue water population is now associated with a constant pore size. This is the reason why they are no longer labeled as capillary pores, but are described as a new pore, the interhydroid pores. As sketched here, it is assumed that these are located between the CSH needles visible on micrographs. The pore sizes of gel and interlayer pores also remain constant. Looking at the amounts of pores formed, one observes that the population of gel pores has reached a plateau, whilst more interlayer pores are formed. At the same time, the interhydroid pore water population continues decreasing. The conclusion is that new CSH is formed in the interhydroid pores, therefore decreasing the interhydroid population. Furthermore, additional layers of CSH seem to form within the gel pores, compensating the formation of new gel pores by filling up the interhydroid pores. This leads to the plateau of the gel pores. For very long hydration time, as soon as the interhydroid water has been consumed, the gel pore population decreases whilst the interlayer population continues increasing. This supports the previous interpretation of the plateau. With increasing hydration time, the gel pores are filled with additional layers as shown here. This must lead to a densification of CSH, which will be discussed on the next slide. In the video NMR-II analysis, it was shown how a system of mass, volume, and molar balances of the constituents of cement can be used to obtain further information from an NMR measurement. This analysis, as explained there, requires further input from XRD and chemical shrinkage measurements. The additional properties which can be derived are the CSH density, denoted ρx. In fact, 2 different CSH densities can be written down. The CSH solid density, referring to the density of the tobermoryte-like layers, and the interlayer water as shown. The second density is the bulk density. It is the density of the CSH layers, the interlayer water, and the gel pores. The color code is green as used later in the graph. We expect that this density will be lower than the solid density since the water in the gel pores has a lower density than the CSH layers. Secondly, we can also obtain the total formula of CSH, which reads calcium Z, silicon Y, aluminium 1 minus Y with X water included. Z denotes the calcium to silicon and aluminium ratio, Y the silicon to silicon and aluminium ratio, and X the water content, expressed as ratio to silicon to aluminium. Similar to the density to water contents, X can be written down. X in red denotes the composition if only the interlayer water is included. X prime in green denotes the composition if also the gel water is included. Let's look at the density of CSH first. In this graph, the x-axis shows the degree of hydration, whilst the y-axis plots the density of CSH. The red curve is the solid density, which remains approximately constant. The slight decrease towards the end is almost within the error bars of the measurement. The bulk density, shown in green, increases considerably with advancing hydration. On the previous slide, we have already seen that the gel pores seem to be filled by additional layers as cement hydration progresses, as shown here. This reduces the volume fraction of gel pores and increases the volume fraction of denser tubermoride-like sheets, which leads to an overall increase of the density. This picture offers an alternative explanation for the slight decrease of the solid density, shown in red. The more layers are formed, the higher the average layer stack height becomes. Therefore, the definition of the solid density includes more interlayer water, which has a lower density than the layers and decreases the average density. In the next diagram, the two CSH densities are plotted against the water-to-cement ratio after 28 days of hydration. The solid density, again in red, is constant within the error bars of the experiment. The green bulk density, in contrast, decreases with increasing water-to-cement ratio. If more water is initially present, more capillary pores are available to be filled by CSH with a high bulk volume. Thus, the bulk density is reduced. We now look at the CSH composition and its changes during hydration. First, we look at the calcium to silicon and aluminium ratio, Z. The x-axis denotes the degree of hydration, the y-axis denotes the calcium to silicon aluminium ratio. It remains constant at first at about 1.8 and decreases then to a value of about 1.7. This could be attributed to the formation of an outer product followed by an inner product, where the latter would have higher silicon and aluminium content. Certainly, the change is not surprising, since the fast-reacting C3S in the system releases calcium and silicon at a 3:1 rate, while the later reacting C2S releases calcium and silicon at a 2:1 rate. We now look at the water content in CSH as a function of the degree of hydration. On the x-axis we plot again the degree of hydration, on the y-axis x and x prime, where the former is the water content, if only the interlayer pores are taken into account, plotted in red, while the x prime, plotted in green, is the water content if the gel water is included. The red solid water content remains approximately constant, which does not surprise since it has already been found that the density is constant. When the gel water is included, plotted in green, the water content decreases with time. This is due to the increase in density by consuming gel pores and filling them up with additional solids. Let's summarize what we have learned in this video. We followed the hydration of white cement in time. Proton NMR enables us to follow the development of the different pore space populations during hydration. The most notable result is the shrinking of the capillary pores towards a constant size before they are consumed, and the progressive filling of gel pores by further layers of CSH. This leads to a densification of CSH and to a decrease of the bulk water content. Also, change in the calcium to silicon aluminium ratio was reported over time.